All right, good morning. Welcome to the session on driving cell therapy into solid tumors. We have a great panel here today. I'm Angela Shen. We're gonna have our panelists introduce themselves as they start to answer their questions. So the first question we have, with regard to CAR T therapy or other types of cell therapy and related modalities, there have been a lot of work done specifically targeting solid tumors. So our first question is, what are the approaches that you and your companies are taking to target solid tumors? What has or has not worked and why? And how are you working to overcome some of the scientific challenges that are associated with these? If I could start with Corinne.
Thank you. Yes, I'm Corinne Goff. I am President CEO of Imunon, which is a small biotech company that is focused on developing a non-viral DNA platform. So it is not cell therapy, but as we'll certainly talk about later, it certainly can be used in combination. We are focusing on immuno-oncology and infectious diseases. And to answer your question, in oncology, our focus is to modulate the tumor microenvironment. I think that's what we are all trying to do, in fact. Obviously, as you know, this tumor microenvironment complicates all aspects of oncology therapies in solid tumors, creating the exhaustion of the C cells. And our approach is to, actually we have three modalities, is to look at, can we stimulate the tumor microenvironment with the expression of cytokines? Or can we go at it with cancer vaccines, neoantigen cancer vaccines, or tumor-associated antigen cancer vaccines? And we are looking at it. So I think I'll have more time to discuss where we are with our lead program, which is an IL-12 expression program. But this is what we are trying to do: is to stimulate to revitalize the tumor microenvironment, so that the immune cells, effector cells, can be effective against the tumors.
Nice, Ryan.
Yeah, so I'm Ryan Richardson, Chief Strategy Officer at BioNTech. At BioNTech, we're a multi-platform company with deep roots in immuno-oncology. In all of our cancer programs today, we have 20 in the clinic across a range of different modalities. All of them are focused on solid tumors today. And cell therapy is an important part of what we do. We also work in mRNA and other modalities too. But within the cell therapy space, we've got a couple of different platforms that we're taking forward. One of the key themes for us is individualization of cancer care, and a couple of our cell therapy platforms enable us to pursue multi-targeting, which we think is going to be a key requirement to really take cell therapy to the next level in solid tumors. So those platforms include more traditional CAR T cell therapy, which is the most advanced, but also personalized TCR. We have an active program there, and also ex vivo T cell therapy, which uses PBMCs to create a TIL-like product based on a neoantigen selection, similar to what we do in the cancer vaccine space. And if I just take our lead CAR T cell program, which is the most advanced, I mean, most of these platforms are still early, and there's a lot of work being done at the company right now to try to bring together disparate technologies, which we think is going to be needed to really take cell therapy to the next level. But just to take that lead program, we're really trying to address two fundamental things with our Claudin-6 CAR T cell therapy, which is, we just had some data at ASCO a week or two ago, and it's shown very, very encouraging early results in the treatment of solid tumors. And those two factors are novel target. We think the field has generally lacked good targets to address in solid tumors. And the second is to try to address the persistence challenge, which seems very frequent. If you look at across the field, almost every study in the solid tumor space highlights a persistence challenge. And so here what we're actually doing is taking a combination approach where we're combining a CAR T cell therapy, an autologous CAR T cell therapy, targeting a novel antigen and combining it with an mRNA vaccine, which we administer post-cellular infusion in repeated fashion and basically boost the CAR T cells in vivo. And I mean, it's still early, but I think we're seeing really, like I said, really encouraging results at the second dose level in our dose escalation study. We're seeing above 50% response rates, and this is exciting. I think we still have to see how the durability looks, but and I'm happy to talk more about that. But I think it's combination therapy is the other angle that we're pursuing.
Well, I want to thank the organizers for having me here. I'm Lynn Seely. I'm the CEO of Lyell Immunopharma, and Lyell is a publicly traded clinical stage cell therapy company. We're focused on solid tumors, and we're approaching this in two different ways. We do have a targeted CAR T cell program. We also have a tumor infiltrating lymphocyte program. And so what we do is we take the patient's own immune cells, we send them to our manufacturing center, we reprogram them both genetically and epigenetically, expand them, and then give them back to the patient. So they have many more of their own immune cells capable of killing cancer. We are currently in 2 phase 1 clinical trials. Our scientists have developed 4 really innovative platform technologies that are stackable, that we use in combination to really accomplish 3 things, which we think are going to be critical to help cell therapy be successful in solid tumors. And of course, the first and foremost is we need the right potency and cytotoxicity, the ability to kill cancer cells. Second, and very important to Lyell, is we need to have T cells that can overcome exhaustion. And I'll describe to you some data that we really believe suggests that this is one of the fundamental barriers that has to be able to be overcome so that the T cells can continue to function in the hostile tumor microenvironment. And then finally, the cells need to have the right phenotype. They need the right durable stemness so that the cells can persist and self-renew and really fight the cancer over time. I think we all know that cell therapies have become really established and very successful in hematologic malignancies, but we haven't seen the same success in solid tumors. And so why is that? I think there was a very elegant clinical trial that was run at the Fred Hutch Cancer Center where Jennifer Spacton, Stan Riddell, and colleagues took a ROR1 targeted CAR T, autologous CAR T cell, and infused it into patients with chronic lymphocytic leukemia, heme malignancy, and saw the expected results. The cells expanded, they persisted, and they helped to eradicate the cancer cells. When they took exactly the same autologous ROR1 targeted CAR T cell and gave it to patients with ROR1 positive solid tumors, triple negative breast cancer, and non-small cell lung cancer, the cells didn't expand, they didn't persist, and they didn't help the cancer. And what they saw is very rapidly the infused T cells developed markers of T cell exhaustion, for the first time really showing the difference between heme malignancies and solid tumors, and that overcoming T cell exhaustion was critical. So that really is one of the key things Lyell is focused on and is taking advantage of in our phase 1 clinical trials.
So you talked about some of these scientific challenges. What do you think are the most ideal ways as you sit here today to try to overcome some of those challenges? And maybe we could start in reverse order this time with Lynn.
Sure. So we're doing this in a couple of different ways. As I said, for us, our focus really is on overcoming the barrier of T cell exhaustion. And so we have a phase 1 clinical trial where we're enrolling patients with ROR1 positive triple negative breast cancer and non-small cell lung cancer. We have a ROR1 targeted CAR T. But the target is important. What we really believe is more important is getting the T cell function right. And so to help the T cells overcome exhaustion, we're genetically modifying them by overexpressing c-Jun, which is a protein that modulates the AP1 transcription factor pathway. It has been shown in Crystal Makel's lab and others repeatedly to help the T cells overcome exhaustion. So we have a targeted ROR1 CAR T cell that overexpresses c-Jun to, we hope, help the cells with overcoming exhaustion and therefore better infiltrating into the tumor. In addition, we use a very special manufacturing protocol, something we call our EpiR technology, which as we expand the cells, we drive them into a more stem-like state so that when we infuse them back, the patient has lots of cells, some which are ready and capable of cell killing, but others which are more stem-like and can have self-renew and persist over time. And so we're trying to overcome this with these two technologies, c-Jun overexpression and EpiR technology. And in our TIL program, this is particularly important because when you're making TILs, and we have a second clinical program that's enrolling now for our TIL program where we're looking at advanced melanoma, non-small cell lung cancer, as well as colorectal cancer. And with TIL therapy, what we've learned from those patients with advanced melanoma who have responded historically at the NCI and other places is that if the cells that they receive contain the right amount of stem-like features, those are the patients most likely to develop clinical responses. So in our TIL program, we have this special manufacturing process so that our end-stage product, where we take maybe from the patient millions of cells and have to give back billions of cells, we can maintain a cell population that is not extremely differentiated and leaning more towards short-lived cells, but actually give them stem-like cells that can persist and repopulate over time. So I think this idea of overcoming T-cell exhaustion and having durable stemness to give the persistency and the durability is very important.
Okay, Ryan?
Yeah, I couldn't agree more with Lynn's comments about the importance of T-cell fitness and the limitation that is T-cell exhaustion. I think our starting point is that we believe that multiple edits to the T-cell backbone are going to be needed to really get to a higher level of T-cell fitness. We don't think that 2 or 3, generally speaking, we don't think that 2 or 3 edits are going to be sufficient. And we think that's really important, and that not all T cells are made equal. We know that, but you're seeing that again and again in the data sets. When we compare autologous, allogeneic, iPSC-derived, et cetera, there really is a difference in T cell performance, and so we think that we have to get to the underlying cause of that. I think the point that I had mentioned very briefly in my opening remarks, another way that we're trying to address exhaustion is through this engagement of the immune system, even with other modalities. And so I mentioned the RNA vaccine as one way to do that. It's a novel approach where we stimulate the immune system repeatedly with an mRNA vaccine that encodes the same antigen that the CAR T cells are targeting. And this is particularly important, we think, in the solid tumor sphere, because unlike liquid tumors where the CAR T cells, once infused, they have almost repeated continuous interaction with antigen, and that forestalls exhaustion. In contrast, in the solid tumor space, a lot of times you don't have that, especially with cold tumors that might be isolated with lack of blood flow, lack of access. And so we're seeing that exhaustion problem even more acute in the solid tumor field. And by using an RNA vaccine, we think that we can effectively present antigen throughout the immune system on dendritic cells. In this case, we're encoding a full-length antigen, the Claudin-6 that I alluded to, and we are seeing evidence now in humans. We saw that in animals, and now we're seeing first evidence in humans that does appear to be stimulating the CAR T cells in vivo and increasing not only the number of active T cells, but early evidence suggests also that it seems to be having an impact on exhaustion and persistence.
Okay, that's helpful. And then Corinne?
Yeah, so our approach is an in vivo approach with the utilization of a DNA that will code for a cytokine, for instance. And our program, which is currently in phase 2, is in ovarian cancer. The difficulty of those tumor types like ovarian, colorectal, that they are very immunosuppressive. So you really need to use a cytokine that have an effect across the entire tumor microenvironment, and that's why we chose IL-12. Now, the difficulty of IL-12 is the PK/PD. There were approaches in the past that were looking at the IL-12 protein as a therapeutic, and that failed because of the very complex pharmacokinetics. So there was accumulation of IL-12, systemic side effects, and this was not the right approach. So we decided to code for IL-12 in the tumor microenvironment. It's an intraperitoneal injection. And what it does is that because it is a DNA expression of the IL-12 antigen, you have like a sustained production of IL-12, which remains local, and we don't see any systemic side effects. So that's an approach that we think is interesting as an in vivo approach to, revitalize the tumor microenvironment. The other approach is similar to what Ryan has mentioned, is we are also looking at a cancer vaccine approach. DNA, mRNA, nucleic acids, kind of the same principle, coding for either neoantigens, so for a personalized approach to the treatment of cancers, or coding for, CAR antigens also is possible in the same way. Now, the difference might be on the prolongation of the antigen secretion. I think what we see with DNA is that the expression of the antigen doesn't peak as high, but maybe is longer, and this durability can be beneficial in some cancer types.
So just as important, but also discussed in parallel, are the operational challenges that need to be overcome in order to tackle solid tumors. For example, the market size of many solid tumor indications being considerably larger than some of the heme malignancies that have been tackled. Could you comment on what your thoughts are on the operational challenges that you see, and if there's anything in particular you're wanting to prioritize to be able to address that challenge? So I'd love to start with Lynn.
Sure. So, the fact of the matter is that solid tumors make up 90% of cancers, which is the leading cause of death, really now starting to overtake cardiovascular disease. Heme malignancies probably make up 5%, so this is going to be a huge change in terms of making these treatments accessible and available. But I continue to believe this number one most important thing in solid tumors is understanding the biology, because once we really understand the biology and get these cell therapies to work, then we can start to really optimize and move them forward. And so we're starting with autologous therapy. Specifically for that reason. We think it's the most likely to work and the most able to help us understand the biology. And then, of course, people will be very interested in taking that and looking at allogeneic and other sorts of modalities. So number one is understanding the biology so we can get these therapies to patients with solid tumors. And then, like all innovative things, there are many, many opportunities and challenges, and the manufacturing time and accessibility is critical, and I think we'll be very focused once we have safe and effective therapies to really get the manufacturing as quick as possible, and then as sort of, shall we say, closed system automated as possible, and so it's much more cost effective.
Yeah, I think there's very big differences economically across the different modalities that we've been talking about. And I think if, obviously, autologous has a very different cost structure than allogeneic from an industrial perspective. I think autologous, from my perspective, is a modality that really, to be highly profitable one day, would need to be done at high scale or with extreme efficiency. And capacity utilization management would need to be really, really strong. We've seen a couple of the leading players in the heme space start to master that, and then you've seen some big differences across the players in terms of those who've done it better than others, perhaps. But I think the limitation right now to getting to scale on autologous is we don't have enough autologous products to actually, with targets that can actually go the distance. We've got a couple of shining lights in the field that I think are very promising, but no mega blockbuster on the horizon, at least that I think that the world can see yet. Hopefully that changes. I think allogeneic, very different, much easier to make money there. Cost of goods is an order of magnitude lower than autologous, but it's more about biology and the technical challenges of making allogeneic actually work as well. So we'll see if the field can tackle that. I think if it can, then I don't think the economics would be very difficult there. One additional area or sort of future pillar that we're also looking very closely at BioNTech is actually in vivo therapy, in vivo delivery of cell therapies, which we think could really turn the economic model on its head if you can master a number of technological challenges, and I think really deliver the sort of a vision of multi-target therapy on a totally different cost structure than you could do with autologous. That's kind of high in the sky thinking at the moment. I think there's a lot of interesting technologies out there. It's not yet been mastered or all pulled together, but I think if that can be accomplished, I think we'd have a very different economic structure that could allow us to tap those markets, perhaps with many different products or variants of products, in a way that was still economically feasible.
And Corinne, your company's pursuing or developing DNA vaccines. Are those operational challenges unique to, say, cell therapies that need to be manufactured?
Yes, I think, to the off-the-shelf approach, if you target tumor-associated antigens, is certainly more cost-effective than an individualized approach. Now, does it work as well? That's the question. But I think, as you look at targeting solid tumors, which maybe are 10 times more, patients with solid tumors than liquid tumors, you need to find economy of scales, and I do believe that the personalized approach is certainly the most sensible in terms of effectiveness, because we have not really cracked the off-the-shelf approach yet. That being said, we also need in the future to have many more hospital centers and manufacturers working on making the process faster and more efficient. And I do believe that's possible, but with every individualized approach, and that includes the personalized vaccines, you still need to get the patient cells and expand the cells and then re-inject. And this process takes time, it's expensive, and we definitely need to work together to simplify as much as we can, because that will be limiting in terms of access if we cannot get to a more simplified way of treating patients.
Okay, so maybe let's move on to some of the challenges that you just described, to how we adapt this to the end users, which are the healthcare providers. So how do you think those healthcare centers should best prepare in order to deliver these therapies to the maximum number of people? And how have each of your respective companies maybe been trying to help with that process ahead of time? So we can probably start with Lynn and go down the line.
Sure. Cell therapies and any innovative new treatment requires setting up new processes, tremendous communication and problem solving. And I think back to the days when bone marrow transplant was new and how difficult it seemed. Now, of course, it happens very, very easily and routinely. With CAR T-cell therapy, for example, in the heme malignancies, when it first came out, it was very frightening and done on a very limited basis. But now the academic medical centers and others are set up with their cell therapists, their care teams, with their processes and procedures, so they do it very easily. They handle the toxicities and side effects with ease compared to the great degree of difficulty at first. Now we're moving into a new arena of cell therapy for solid tumors. And while medical oncologists, we find are, of course, very comfortable with immune checkpoint inhibitors and other immune therapies, they haven't done much in the way of solid tumor cell therapy. And so this now requires a new skill set where the cell therapists that have been focused mainly on hematologic malignancies and their care providers need to partner with the solid tumor oncologists who are caring for these patients and need to be able to talk to them about the trials and encourage them to enroll in the clinical trials. And so there's a whole new set of communication paths and patient referrals and care plans that need to be developed. With our TIL therapy, it's even more higher degree of difficulty because there the surgeon has to take a tumor sample to send to collect the tumor infiltrating lymphocytes. And so yet again, a different set of communications. But there's phenomenal opportunity because those academic medical centers who are sort of early to set up these systems of communication and processes can really be the leaders. So we spend a lot of time educating, helping to build bridges and set up communication channels within the centers. And I think it's very promising, and over time people get more comfortable and it becomes simpler. But anything new is always has some challenges and barriers.
Okay, that's helpful. And then what's your take on it, Ryan?
Yeah, I mean, I just to second some of those points, I think it's going to require capability enhancement, obviously from industry on the company level, but also more so from the providers of the hospital than for traditional pharmaceutical products. There's no question about that. And I think actually the interplay between the two is important. You know, when you think about the go-to-market model for a cell therapy from a company's perspective, you're really thinking about dedicated site personnel that are focused on not only having a relationship with the sites, but manage the logistics all the way to the patient. It connects basically the site all the way back to wherever the product is being manufactured, right? And so currently that's mostly within companies. In some places you actually also see some manufacturing at the site itself. More in Europe, but you see that in a few places. But I think that means you have to have this sort of straight line connectedness or connectivity, and that's logistically a challenge. There's no question about that. I think the other point I would make is that to really take this to the next level, I think once we have the products and the data to support them, then I think we're going to need more than just the high-end, certain subset of academic medical centers. Really going to need models that can really extend across the whole healthcare system.
And then Corinne.
Yeah, so there's one aspect also in all the technologies that we are talking about, whether it's an in vivo or ex vivo technology or a combination of both. These technologies are very data intensive, and a key challenge is to get access to the tumor and to get the biopsies, because we need the translational data to understand whether or not it's working, right? If the IgG12 is being produced, if the T cells are revitalized. So it's a consideration that is very important. It is not so essential when you have other therapeutic options like the monoclonal antibodies, for instance. But in T cell therapies and in DNA, mRNA approaches, access to the tumor is important. That's the key to the personalization.
Building off of that point, access to tumor, the need for tumor biopsies, measuring disease response in solid tumors has its own unique set of challenges as compared to measuring a tumor in hematologic malignancy. In terms of getting access to tissue biopsies, are you finding it difficult to get serial biopsies? How are you doing that? How are you sort of mitigating also some of the challenges that come with radiologic interpretation of disease response, which can also be challenging when it comes to immunotherapy in oncology?
I'll start with a brief comment. It's actually maybe more relevant, my answer, to what we're doing on the cancer vaccine side, actually the personalized cancer vaccine, but it's got some similarities to cell therapies. I mean, we've actually found that in our cancer vaccine trials, and here I'm talking about our personalized neoantigen-based RNA vaccines, that actually the tumor sample matters a lot. And we need that up front, not because we need to harvest cells, but because we need to do the whole genome sequencing on it to identify the mutanome or the mutation signature of the tumor. And we've just found over the course of having now been in the clinic for almost 10 years that early days we were having a lot of dropouts because the tumor sample wasn't sufficient, or we were not getting a consistent read or a reliable read. And actually we kind of traced that back in some cases to just how physicians or how certain institutions were actually handling samples, maybe letting them sit for a while before they were shipped in. That we found out that could have an impact on the mutanome reading, et cetera. So actually, it's been a really hands-on experience for us in the clinical setting to try to teach sites, first diagnose what the issues and constraints are, and then spend time with sites, try to get them to change certain practices so that we can have a more efficient readout, lose fewer patients.
Yeah, just to build on that, I mean, and what Corinne said is the translational science is so critical here because we're learning. And as we've talked earlier, if we don't get the biology right, we're not going to be successful. And so each time we do these clinical experiments, we have to learn what's working and what's not working. And the best way to do that is with tissue. And so when you set up these trials, it's incredibly difficult. And I'll just use our TIL program. You have to have tissue to resect for the initial product, or in our CAR T program, you have to know that the patient has the right target, the ROR1 protein, that takes tissue. And then what you need is to have the right tissue, the right target lesions to measure over time to assess response. So you've got to have that. Plus you want areas to biopsy because you want to be able to understand before and on study. You can look at things, for example, we have a very robust translational program because we're trying to understand, are we in fact helping our T cells resist exhaustion? Are we in fact getting the expansion and the persistence that we want? And the best way to do that is with tissue biopsies. And so it's enormously difficult to both educate the sites, set up these translational programs, and all the communication and training that it takes. What we've been finding though is when you do that, it's hugely helpful in understanding what's happening, and that the patients, quite frankly, even though it's a tremendous burden for them, are willing to do that because so many patients with late-stage cancer right now want to be able to contribute. And so we have been quite successful in getting the tissue that we need. We too have very hands-on approach to making sure we're getting the right tissue and that it's handled properly. And then with respect to measuring response, we all know that response in solid tumors can be difficult. And with immune therapies, it can be even more difficult because you have this concept of pseudoprogression, where because you get immune cell infiltration into the tumors themselves, they can look like they're progressing early on when in fact it's actually a sign that the products are working. So again, like all amazing transformative therapies, there are challenges that need to be overcome, and that's what makes this fun and so rewarding. So all of these things we have to pay very careful attention to and work collaboratively together. Companies have to work with the sites and with the patients, everybody together, trying to make this work.
What are some of the things you can do or put into place to make it easier for the patients and clinicians to be able to engage in the serial tissue biopsies?
I think it depends a lot also on where the patient is in terms of progression of the disease. Our program, which is in which is used in first line, so in combination with chemotherapy, and then we have a program starting with bevacizumab. But when you take a patient first line and maybe they have a first course of chemotherapy and then they go through debulking surgery, the debulking surgery is the opportunity for the surgeon to take a sufficient amount of biopsies, and that's what we need. And it takes a lot of training of the centers to understand how much tumor you need. So it's certainly easier in first line therapy than in the later lines. But it's at the end of the day, and I think to Lynn's point, it takes trial and errors and teaching the centers how to get to the optimal results with the pathologists and determine the conditions of storage of the biopsies and tissue samples as well.
Great. I think we have time for one more question for now. in terms of, there are a lot of we're seeing different cell types now being engineered. We're seeing T cells, NK cells, macrophages, B cells, et cetera, different cell types being engineered. In the human body, obviously, the cells are a gamish, they're mixed. But in terms of drug development, we're seeing one cell at a time being interrogated, whether it's a T cell or a different type of cell. So just thoughts on why do you think that's happening? Is that optimal? And do you think eventually the cells are going to be reunited?
Well, sure, I'll take a stab at that. It's a great question. And I mean, the human immune system is incredibly powerful and has evolved over an enormously long period of time. So of course, each cell type is important. Our company, Lyell, is focused on T cells because there's a tremendous amount known about T cells, and they are central to cancer cell killing. Our hope in focusing on T cells is that we get in there and can actually, with our CAR T cell program, for example, start the cell killing, that then actually the tumor can open up, the microenvironment can flip, and the other cells can come in and sort of assist and help. I think, of course, also companies in general pick a cell type to specialize and focus on, because the more variables you introduce into any scientific experiment, the more likely you are to fail. So asking one question at a time is important. But that being said, in the future, as these different cell types come up, could I see them working together? Absolutely. The same way we're likely to see combination therapies, and we have seen in oncology combination therapies being better than monotherapy. Different cell therapies and different cell types could also be useful together. So it's going to be really fun. We're at the tip of the iceberg here. It's going to be really fun to watch this field evolve.
Yeah, I couldn't agree more. Our focus primarily is the T cell as well. And we still think that T cells, we haven't cracked that case yet. We have the T cell optimization. There's still clearly low-hanging fruit, or at least room for improvement, substantial improvement. So that's our focus.
Wonderful.
Okay.
Any other? Yeah. Maybe since we have a couple minutes left, I'll throw in this last question. So we've spent a lot of time in the last few minutes talking about the specifics of solid tumors and drug development for those tumors. And one question I have for all three of you is, do you think that combination with different modalities could increase the chances of being successful in treating different solid tumors? So maybe we can start with Corinne on that.
Yes, I absolutely do believe that combinations are key in the treatment of solid tumors. I think CAR Ts now are a mainstay, right, in the treatment of cancer, and will make it in solid tumors, I have no doubt. I think the approach that Lyell has is very, very smart. But to combine with a cancer vaccine makes a lot of sense so that you can, increase maybe the engraftment of the CAR T cells and limit the exhaustion. And then, of course, combination with PD-1s or PD-L1s, is a given. So it's an important consideration as well from economic point of view, as well when you start thinking of the number of therapies that the patient will have to take, and first the cost that is an important component, but also how do you combine, right? What do you administer first? What comes second? What makes sense? And we have a lot to learn about the most efficient combinations and how to go about it.
Ryan?
Yeah, we agree with that. We do think combinations are going to perhaps even be needed to really make a big difference in cell therapies across different tumor types. I think, so I mentioned cancer vaccines and CAR T-cell therapy. We're also looking at TCRs, and also potentially vaccines with TCRs could be interesting, but also more traditional combinations like checkpoint inhibitors. We have a class of medicines called ribocytokines, which are RNA-encoded cytokines, which we think also could be an important combinatorial agent, and others too, depending on the tumor type. We think we need a multimodal approach.
Okay.
I guess the only thing I might add to that is, as Corinne sort of alluded to earlier, is getting these cell therapies as early line as possible. I'd love to see them used up front in combination with surgery, radiation. We're forced by our development process to look at relapsed refractory late-stage patients at the beginning, well, those are the patients the least likely to respond to immune therapy because they've already been through so much. And so the sooner we can get these treatment earlier line and sort of used early in combination, I think the better off patients are going to do and these therapies are going to perform.
Okay, perfect. With that, we're out of time. So thank you for joining us this morning. We really appreciate it. And thanks everybody for listening.